Hello, I'm Mikhail Shapiro. Nice to meet you guys over this interface. I'm a professor of chemical engineering, and I'm going to tell you today about some of the work that we do in our lab, and here are some of the folks who are doing the work, and I hope some of you will join us. So the work in our group is focused on developing technologies to image and control the function of cells deep inside the body. We want to do this for a couple of reasons. First of all, for basic biology, because no matter whether we're talking about neurons in our brains or microbes in our guts or immune cells looking for tumors elsewhere in our body, we want to study how those cells work, not just under a microscope on a slide, but within the context of the intact organism and all the tissues and cell types and physiology that's happening there. And we need ways to interact with those cells when they're inside the body. In addition, we and other people are interested in developing cell-based diagnostic and therapeutic agents. And some examples of that recently include immune cells that have been engineered to go into the body of a cancer patient and detect tumor cells and kill them, or probiotics that are now engineered that you can eat in your yogurt and that will go through your GI tract, detect inflammation, release anti-inflammatory compounds. And these are very powerful agents, but right now, after we put them into our bodies, we don't have very good ways to tell where are they. Are they alive? Are they executing the functions that we programmed into them? And don't have good ways to tell them what to do based on their location. Now, the reason that's challenging is because if we think about the technologies available to image and control cells inside the body, the best tools we have are things that use light. These are things like fluorescent proteins or optogenetics, which are proteins whose function can be controlled with light. And they derive a lot of their power from the fact that they're genetically encodable, meaning that we have very good ways using modern synthetic biology and genetic engineering to plug them in intimately to what's happening inside of cells. But the problem is that light doesn't penetrate very deeply into our body. It gets scattered by tissue within about a millimeter. And so as a result, these technologies are difficult to scale to intact organisms. At the same time, we have technologies like ultrasound and magnetic resonance that use more penetrant forms of energy, meaning that sound waves and magnetic fields, by and large, can get in and out of our bodies no problem. But these technologies have not been connected as well to what's happening at the cellular and molecular level. And so what we try to do in our lab is to take the physics of sound waves and magnetic fields and take these technologies and extend their capabilities down to the cellular level through biomolecular engineering, meaning that we find, we engineer, we evolve proteins and other genetically encodable materials that have the right physical properties to, instead of interacting with light, to interact with sound waves and magnetic fields. And the work in our group roughly breaks down along the lines of sound waves and magnetic fields and designing ways to use them to image or control the function of cells. I just want to give you a couple examples of that to give you a flavor, focusing on the ultrasound side of what we're doing. So you've certainly heard of ultrasound imaging, and for the vast majority of us, our first picture was an ultrasound picture when we were inside of our moms. And it's a very simple technology. It works by having sound waves that move through tissue. They encounter an interface with something that has a different density or stiffness, and then some of that sound wave gets reflected back. And based on when that signal comes back to our transducer, we can tell how deep that object is. And we can sweep the sound wave around and get an image like this one. Now, everybody knows about this kind of ultrasound, but what people may not realize is just how high performance a technique it is. So easily in our lab, without any special equipment, we can get spatial resolution centimeters deep inside the body that's better than 100 microns, which is enough to see many important tissue and cellular scale processes. It's also very fast, faster than a millisecond. Now, modern ultrasound is also very portable. You can get an ultrasound system for $2,500 that plugs into your smartphone. And recently, people have also developed super resolution ultrasound, where what you're looking at in this case is an entire rat brain and cross-section, and you have the microscale blood vessels within this brain resolved at 7 micron spatial resolution inside the body. So that's fantastic. But of course, here I'm just showing you anatomy, and what we want to do is connect sound waves to what's happening inside cells. So how do we do that? So here, nature really gave us a gift. And this gift came from cyanobacteria, like the ones pictured in these bodies of water, that had to solve the evolutionary problem of regulating their buoyancy, meaning that they needed to find a way to float to the top of the water so they can get enough sunlight for photosynthesis. And the way they evolved to do that is by forming these really beautiful, self-assembled, intracellular, air-filled protein nanostructures called gas vesicles. This is a picture on an electron microscope of one of these structures that we purified from cyanobacterium. And what's special about these things is that they're a couple hundred nanometers in size. They're made of a protein shell that is hollow on the inside and filled with air. So you have this very thin shell, and the way this works is that air can come in and out according to its Oswald coefficient or solubility, but water is prevented from coming in and condensing into a liquid on the inside. And this is actually a pretty incredible piece of evolution, because if you ask a chemical engineer to make you a stable nanoscale bubble, they'll say, I can't do that, because by the Young Laplace equation, the smaller you make a bubble, the greater the pressure differential and the less stable it becomes. But nature figured out how to do that by making essentially a solid shelled structure rather than a surface tension type of mechanism. And these proteins are encoded by a set of genes inside of the cyanobacteria that encode some structural components or assembly factors or chaperones that bring these proteins together. And when these cells want to float higher, they turn on these genes to make more of them. And when they want to sink, they can break them down with proteases and sink back down. So we looked at this and we said, okay, well, here is an object that a cell can make based on a genetically encoded sequence that will have a different density and stiffness relative to its surrounding medium, which means that it might be able to scatter sound waves and thereby be seen by ultrasound. And indeed, that turned out to be the case. So this is looking at some of these proteins that are purified and embedded in a hydrogel, which is acoustically transparent. And we can see very nice ultrasound contrast in this case. And of course, one of the first things we wanted to do after we see that is inject this into a mouse and see if we can see it within the context of a living animal. And so here, we're going to be looking at the liver, which is outlined here. It initially starts out being dark. And as we're injecting these nanostructures into circulation, it gets into the liver, and you can see the liver lighting up very nicely. So we were very excited about this when we first made this discovery, because this is the first protein that could be seen with ultrasound. It's similar to when GFP was first found in jellyfish as a fluorescent protein. And so over the last few years, we've had a lot of fun working with these proteins, because now we have this unique biomolecule that has mechanical properties. So we look at how it responds to pressure, and it has these buckling and unbuckling interactions that produce unique sound wave signatures that we can design custom ultrasound pulse sequences to visualize. We can also play around with the different proteins that are part of these gas vesicles, as they're known, and change the mechanics of that shell, or cause them to get larger, get smaller, change their shape, and all of these things have an impact. Now, one of our biggest challenges that I want to say a little bit more about has been how to take this relatively complex genetic program and to move it into other kind of species that we want to image, right? We're not particularly interested in cyanobacteria, but we want to look at things like immune cells and cancer cells and neurons and commensal bacteria in our guts. So one step we recently took that was quite a bit of work that actually people weren't sure we would be able to do, is to design a mammalian version of that multi-gene operon that allows the expression of these gas vesicles in mammalian cells. And so several students in our group worked on this problem and designed a way by borrowing components from these cyanobacteria, some viral elements, and stringing these genes together in a way that when you put them inside of a mammalian cell, you can get gas vesicles formed inside that cell. And now you can use ultrasound to watch gene expression inside of, in this case, a tumor. So there's a lot of work that's happening on this imaging side. But one of the things we've learned just in the last few years that's driving some of our future research is that everything I just told you about with ultrasound is in one little regime of ultrasound, where we're sending a pulse and we're listening for its echo to help us with imaging. But actually, ultrasound can do a lot more. It can also push on things with sustained pulses and create a force on objects or cells. It can heat things up, or it can create bubbles and cavitate those bubbles to unleash strong mechanical forces. And so I just want to give you two examples of that very briefly. So one thing we found is that we can use ultrasound to acoustically manipulate cells. So here we have bacteria that are expressing these acoustic biomolecules, these gas vesicles. We put them into a microfluidic channel, and we apply ultrasound so that there is a pressure maxima at the walls of this channel. And what we found is that by having these gas vesicles inside the cell, they can experience a force due to this acoustic field that in this case will push those cells to the walls of the channel. Watch, watch, watch. Okay, well, my computer is moving pretty slowly. Okay, let's try that again. Okay, well, you can look at our paper and see that for some reason the connection here is not great. Another thing we can do, hopefully this video will work, is that we can create a more complex spatial pattern. So here we have a bunch of cells in a microfluidic channel. We apply ultrasound, and we can arrange them. And then by changing the sound wave frequency, we can get them to dance around. We can also trap them at an acoustic focus and then move them around by moving that focus around. In this case, we're writing out the letters CIT. There we go. We can also use sound waves for remote control of gene expression. So we found that one of the things you can do with focused ultrasound is generate small increases in temperature in a very controllable way. And we've generated a set of thermal bias switches that operate within the safe range of temperatures between 37 and 42 degrees and allow us to turn gene expression on and off in response to that mild heat. And so then what we can do is we can use focused ultrasound with millimeter precision to heat up little regions of tissues or in vitro samples, and essentially use that to paint a pattern of gene expression using ultrasound. We can do that in vitro here and in vivo in a mouse. And then the final thing I want to mention is using the cavitation phenomena is we have a way to control different parts of the brain non-invasively by using bubble formation and cavitation to open the blood-brain barrier, allow the entry of viral vectors into a particular part of the brain that encode receptors that allow us to subsequently control the neurons in that part of the brain using designer drugs. And you use that to control memory formation in mice, and you can read more about that in the paper here. So the last thing I just want to say is, why are we the ones to do this? What business do chemical engineers have doing this kind of research? And the answer is that this is a very multidisciplinary work that requires people who are comfortable with molecules. Here we're engineering and thinking about things at the molecular level, but are also comfortable with physics and math because we're thinking about the physics of acoustics in the case of what I just told you about. We think about kinetics, we think about thermodynamics, we think about transport, we do computational modeling. And so it really requires people who are willing to work across all those disciplines. And if you look at our lab, we have a really amazing team of people that come from all across these different disciplines. They're graduate students from 6 different graduate programs at Caltech and postdocs from all over the world and all over different disciplines, which is really fun for me because it makes for a very rich intellectual environment. So if you're interested in this kind of work, I encourage you to contact me. I'm happy to talk to you about it. Again, I'm sorry that we couldn't welcome you to campus in person, but I hope I'll see you here in the fall.